Hi, I am Neeladri. I'm a postdoc in UCL. Uh, However, I'm going to talk about a work that I did during my PhD with Dr. MS, Professor MS Madhusudan at ISMB Pune. It is about a structural database of chain and domain interfaces of proteins. Why would we like to create such a database? Uh, there currently exists a lot of protein-protein interaction databases such as String and Interactome 3D, etc., but they are mainly sequence-based. There are structure-based databases, but some of them are interchained like ProdKit, and some of them are domain-based like 3D. We wanted to combine both chain and domain interfaces because uh, gene fission and fusion can convert one type of interface to another. And in addition, if we have a combined database, we can identify structural and physicochemical similarity between them. Uh, Recently, a study by Mirabello and Wallner have shown that topology independent matches of interfaces can be used to identify templates to model complexes. Over here is a figure that shows two proteins which are not identical to each other structurally, but the interface is structurally identical. So, and hence, if we have a library of domain and chain interfaces, they could serve as templates to model protein complexes. With this in mind, we wanted to create a database of chain and domain interfaces. So the main objectives was to create a database of domain and chain interfaces in the PDB, to structurally cluster them, to get a redundant set of structural clusters of the interfaces. Also, we wanted to identify common pattern between these interfaces. In addition, we also wanted to identify small molecule binding site at the interfaces, because this could help in inhibition of protein-protein interactions. So the data sets that we use in the study are, uh, we get all the protein structures from the PDB, we get the domain definitions of the protein folds from cath. Uh, Again, our data sets contain both domain and chain interfaces. Uh, our data sets also contain interfaces that are both homomeric and heteromeric. So over here in the lower panel, the first one is a homomeric interface, where the interface is between two chains or domains that belong to the same fold, and heteromeric is the one where the two chains belong to different folds. So for our interface, we only considered cases where the changing axial surface area after complex formation was greater than 400 angstrom square. This was to ensure that we do not have any crystal artifacts in our data set. We ensure that the distance between the residues of two different chains or two different domains were at least 8 angstrom from each other. So our interface definition had a cutoff of 8 angstroms for interface residues. So the library of the chain and domain interfaces contained about 112,000 chain interfaces, which belong to 2444 fold combination. So to iterate what a fold combination is, say we have fold A interacting with fold B in the PDB. So AB is a fold combination. So the total number of chain interfaces that we have, out of which around 62% of the chain interfaces are homomeric, which belongs to 514 folds, and the total number of heteromeric interactions belong to 1930 fold combinations. The total number of domain interfaces that we have is 66,000 about, and they belong to around 28,000 PDBs, and they belong to some 1100 fold combinations. So, however, only 28% of the domain interfaces were homomeric, which belongs to 107 fold combinations, and the number of heteromeric fold combinations that we have in domain interfaces around 1000. So the cath database has about 1391 folds, out of which we have seen around 1109 fold which interact with at least another fold of same or different type. Of these 1391 folds, around 1092 folds interact with less than 30 other folds. We noticed that higher the number of EC terms in each of the fold, the higher the number of interactions it has with other folds. So over here is a plot that shows the number of EC terms along x-axis and the number of folds one particular fold interact with along y-axis, and they have a strong correlation coefficient of greater than 0.8 between the number of folds it interacts with and the EC terms. And the inside figure is just a smaller version of it, which where I have truncated the total number of EC terms at 100 and total number of folds at 102. So if you see the large outlier over here, which has the highest number of EC terms, the Rossmann fold, and it also has the highest number of folds that it interacts with. A couple of other interacting folds that have a large number of EC terms, and the number of interacting fold is immunoglobulin fold, alpha beta plates, Timbels, and jelly rolls that have been highlighted over here in the figure. The next thing that we do is to structurally cluster the interface. So for that, we had to structurally cluster the interface within a fold combination, because if we had to structurally cluster all the interfaces irrespective of the fold combination, it was a lot of computation which is not possible to do. So please bear with me as we go through the schematic. So we have all the interfaces in a particular fold combination in the square box, which contains like rods and circles and triangles. We choose the one with the highest resolution, which is the gray rod, and what we do next is structurally align all of them using our superimposition tool called Click, which is developed by Min in the lab. And we see all the rods are matched to it, while the triangle and the ovals are not. So the first cluster is out, which only has the rods, and next we update the set, and again we reiterate the same process, and we end up with the cluster of a triangle and another cluster which has the ovals. So we notice that 1563 156,000 interfaces belong to some 28,000 clusters. The next thing that I wanted to do is I wanted to see the sequence conservation of the interface compared to that of the whole protein. So we see that over here is a density plot that gives the difference between the interface identity and the full protein identity. So if you look at the right side of the plot is when the interface has a higher sequence identity as compared to the full protein, and the left side of 0 is where the full protein has more sequence identity as compared to the interface, and the red line is plus minus 0.5% of it, where we consider that both of them are equally similar. So we have around 54% of the cases where the interface is more conserved, and around 23% of the cases where the whole protein is more conserved. So from the clustering, what we saw, and the sequence conservation study, what we saw is that around 2% of the interface that were clustered together had a sequence identity of less than 30%, while about 10% of them had an identity of 40%. So over here is an example of a human protein which is shown in orange and golden ribbons, and the bovine protein SC007 which is shown in green and gray ribbons, and the interface is in much darker color, and the remaining part of the protein is shown in a more transparent color. And we see that the two proteins, they have less than 30% sequence identity, but because they belong to the same fold, they are very structurally similar to each other, and as a result the interface is also very structurally similar. We also did some more searches where we try to see if there are structurally similar interfaces across different folds, and here is an example of one such case. So we have the NASG protein, which is in orange, and DNA dependent RNA polymerase, which is in yellow. So they are a complex which interact with each other. And then we have the SPT4 and SPT5, which is in some shade of green and gray. And if you look at the interface, you will see that the interface is very structurally similar to each other. But when you look at the whole protein, you will notice that the SPT4 is not structurally similar to the DNA dependent RNA polymerase E. So they belong to different folds. The DNA dependent RNA polymerase E belongs to ruberithin domain 2 fold, while SPT4 belongs to herpes virus fold, while the NASG and SPT5, they belong to the alpha beta plates. So we have seen that there are cases where the interface is structurally similar, whereas the whole proteins aren't structurally similar. We have also seen cases where there is a structural similarity between the domain and a chain interface. So there are around 514 fold combination which contain both chain and domain interfaces, and we have around 102 clusters which contain both chain and domain interface which are clustered together. So over here is an example of carboxysome cell protein CCMP, which belongs to the alpha beta plates, and the first one is a domain interface. And if you look at the chain interface in the second figure, you will notice very nicely that the two interfaces are very structurally similar to each other. What we also wanted to do along with the structural study is to see if the preference of amino acid pairing uh, is similar between a domain and a chain interface, and we see it's indeed true. So for that purpose we used this uh, uh, potential pizza that was developed in the lab by Abhilesh and Ankit, and we calculated the pairing potential for a domain and chain interface, and over here is a heat map that shows the same. Higher the value, more probable is more likely is the interaction to be seen in the PDB. And we see that the trends are very similar in domain and chain interfaces of pair preferences. However, we see that self pairing is more preferred in a chain interface as compared to domain interface. Again, likely stems from the fact that around 62% of the chain interfaces are homooligomers, while only 28% of the domain interfaces are homooligomers. In addition to this, we also try to see where the binding sites are at the interfaces. So for that purpose, we use this tool called residue depth, which was developed by KP Tan in the lab. And we searched at the interfaces and tried to see how many of them have binding sites. So out of 112,000 chain interfaces, 61,000 had at least 50% of overlap between the interface residues and the small molecule binding site. So if we have a binding site at an interface, the binding site could be inhibited by a small molecule, preventing the interaction to take place. So over here is an example of caspase 9 and B3ZA protein, uh, and we see that the complex can be inhibited if in the binding site inhibitor CO9 is placed. So if the CO9 is present, then caspase 9 can't bind to BIR3. So if we have a data set of all the binding pockets of the interface, this could be used to inhibit interactions between protein interfaces. We also wanted to see what happens with model protein complexes. For that, we have used the model protein complexes that were uh, published by Humphreys using AlphaFold 2 and Rosetta Fold, which contain around 1106 eukaryotic binary complexes. After PLDTT cutoff of good quality and good quality CAD domain assignments, we were left with 308 interfaces. They belong to 233 fold combinations, out of which we only saw some 131 fold combinations in our database. So there were some 102 fold combinations that were not present in the database, which were identified by the deep learning tools. And then what we did was, for the 203 interfaces that were belong to the 131 fold combinations that we have in our database, we tried to see if we could cluster them with our database. So for the clustering, we used two parameters. One was a structure overlap, which gives an idea of how much percentage of the structure has an overlap, and also the RMSD. So for our interface library, the cutoffs that we used was 80% for structure overlap and 1.5 angstrom for RMSD. But with those cutoff, we only matched some 53 model interfaces to our database. But if we allowed a more permissive cutoff of 174 interfaces could be matched, and because it's a model complexes, we might need to be a bit more permissive with our cutoffs. Uh, but however, there are 29 interfaces that could not be clustered, which gave some more ideas of newer modes of interactions that couldn't be seen in the PDB. In addition to this, what we are currently doing is trying to build a interface library web server. So the web server in the web server, one user could give in a PDB, and on the right hand side, if you given a PDB, you could get more the interface residues highlighted in red and blue. The user could also give in a particular fold, uh, wherein with that fold the user can see what are the PDBs that have that fold, and 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 see the clustering of the data in it. uh, We are working on it and we will make it available soon. So to summarize what I have done, we have created a database of chain and domain interfaces, and we have structurally clustered them. We notice that higher the number of EC terms, higher the number of folds they interact with, and also that parts of the work that I haven't shown is the higher is the total number of interfaces that are there for them, and higher is the total number of clusters too. We show that the sequence conservation of the interface is greater than that of a whole protein, and then there are structural and physical chemical similarities between domain and chain interfaces. Similarly, we showed examples of structurally similar interfaces across different folds. We have created predicted small molecule binding site at the chain interfaces, which could be targeted by drugs. So the future of the work would involve like the dataset being used to as templates to model protein complexes. The binding data could be used to develop inhibitors against interfaces. And in cases where there are mismatch between the EC terms and the known interacting partners, might be this could serve as a basis for searching new interacting partners or EC terms. We have around 282 folds which have no evidence of interacting with other folds in the PDB. We can go and see if we know that these folds interact with other proteins in this, from the sequence data sets. Uh, we could also add model complexes to the data set, but we also have to identify at what cutoffs all this clustering and all needs to be done. We are going to surely release a web server to be used by the community soon. To end, I would like to acknowledge my supervisor, my PhD supervisor, Madhu, who was an incredible person to work with. Would also like to thank Gulzar and Ajinkya, who are helping me with the development of the web server. I would also like to thank my lab members from my PhD. And an incredible amount of thanks to my current supervisor, Christine, who was kind enough and great enough to allow me to talk on my PhD work. The funding for this project was coming from DBT Wellcome Trust, and my fellowship was coming from CSIR. To end, we have published it in Protein Science last year. You can scan the QR code and get the paper. You can contact me via Twitter, LinkedIn, or email. Thanks a lot for listening to this entire talk.